Hello, this is a review for your exam 3, Bio 282. My name is Justin. If there's ever anything that I might be able to help with, please don't hesitate to email me, and let's get started. Let's start everything off with the nucleotide. Nucleotides are the monomers that make nucleic acids, namely DNA and RNA, which would be the polymers. Nucleotides are composed of three main parts: a central 5-carbon sugar, a negatively charged phosphate group, that would be this entire thing with the phosphorus and all the oxygens, a nitrogen base, which has a ring structure, and that's it. The structure of the nucleotide is important to know, and we like to think about structure in terms of how components are attached to our central 5-carbon sugar. Before we cover that, let's briefly go over skeletal structure. This is a skeletal diagram, and the carbons and hydrogens are not usually seen explicitly. So a carbon is represented by a band or naked end. So this band over here, there's a carbon there, as along with this one, there's a carbon there. Now this end over here is not a carbon because it is not a naked end. There's an oxygen there. This band or this carbon is bonded to this oxygen. Now that we got that out of the way, earlier I said we like to think about structure in terms of how things relate to our central carbon. So we want to make a map of the carbons within our sugar. And to do that, we label each one of the carbons. We start our labeling off with the carbon that is attached to the nitrogen base. So we call this carbon 1', and we go all the way down to 5'. So going down the line, 1', 2', 3', 4', and this awkward bend over here is our 5th carbon. Now that we got that out of the way, we want to look at what components are attached to what. Attached to our 1' carbon is our nitrogen base. Attached to our 3' carbon is a hydroxyl group or an OH, and attached to our 5' is a phosphate group. This is important to know because DNA and RNA, which are chains of these nucleotides, have directionality, and this directionality is due to this structure that it has. So nucleotides, they differ from each other in their nitrogen bases. DNA can have 4 kinds of nitrogen bases. They can have an adenine, thymine, cytosine, or a guanine. And these are different in their chemical properties, they differ in their structure. And so you can have 4 different kinds of DNA nucleotides. RNA also has 4 of them. You have that adenine, cytosine, guanine, but instead of a thymine, we have a uracil. And nucleotides also differ from DNA and RNA in what kind of sugar they have. For a DNA, on that 2' carbon, it only has a single H. Whereas for an RNA on the 2' carbon has an OH. You won't need to know this for the exam, but it might be useful to know that DNA and RNA nucleotides differ in what kind of sugar base they have, along with what kind of nitrogen bases they can have. So to summarize everything I want you to know about the nucleotide, I want you to know how to label them, what is attached to the 1', the 3', and the 5', that the nitrogen bases can differ for the nucleotides, and we'll cover this in greater detail later on, and also that our phosphate is negatively charged. And because nucleic acids, DNA and RNA, are made of links of these nucleotides, that means that DNA and RNA are mostly negatively charged in function. As we go further, you're going to hear a lot of 5', 3', 5' to 3', and I'd like to cover phosphodiester bonds because I hope that they'll help explain why the directionality is so important, or even why directionality is present at all. So remember, over here, we have a nucleic acid of length of 2 nucleotides. Remember, nucleotides are linked together. They are the monomers are linked together to make up nucleic acids, which are the polymers. And this could be either DNA or RNA, but let's just call it DNA for now. So we have two nucleotides. We have our nitrogen base that is ring structured. We have a nitrogen base that is ring structured. They seem to differ over here, and that makes sense because we can have 4 possible nitrogen bases for a DNA nucleotide. And let's number it. Let's number all the carbons. And remember, we number according to our central sugar, and we start with the carbon as attached to the nitrogen base. So that's 1 prime, 2 prime, 3 prime, 4 prime, 5 prime, 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. Okay, so this matches up with what we know. On our first carbon, we have a nitrogen base. On our 5th carbon, we have that phosphate group, but we see on the 3rd carbon, there is actually a phosphate group attached to this one, not instead of the hydroxyl group. And that is because the nucleotide, while it had an OH in its nucleotide form, but it was not attached to anything, once it makes a bond with another nucleotide, some components are released, namely the OH group. And this bond going from the 3 prime to the 5 prime carbon, the 3 prime carbon of one nucleotide to the 5 prime carbon of another one, is the phosphodiester bond. So let's model what it would be like to make our 2 nucleotide long nucleic acid into a 3 nucleotide long nucleic acid. So we want to add this one on. Now, if you remember anything from our last unit, we talked about about ATP. And ATP has, like this, adenosine triphosphate, has those phosphate groups which are highly negative. It is very easy to break that bond because they're repelling each other. And once you break that unstable bond, energy is released. Energy is released to make a bond or do some kind of work. In this case, we're trying to add this nucleotide onto this one. And what happens is I'm going to, these phosphate bonds are broken and energy is released, and that allows that bond to be created between this nucleotide and the existing nucleic acid string. So I'm going to model it simply. So let's say that we're adding it on. So we have our phosphate group. And also notice how I'm drawing the phosphate groups like this as opposed to the regular structure. You will not need to know it for the exam. I think the most important structural component to know is the structure of the sugar. But you have your phosphate, it's attached to that 5 carbon sugar. You have a nitrogen base. And we're just redrawing it. Yeah, whatever. Okay. But when we're making this new bond with this one, it goes from our 3 prime carbon at this one to the phosphate, this one over here, that is attached to the 5 prime carbon of our nucleic acid that we're adding it on. A bond is made. Yep. So now this is a 3 nucleotide long nucleic acid. Now, we say that nucleic acids, DNA and RNA, have directionality because they go in a particular direction. Over here, on this end, we have a 5 prime phosphate group. And on this end, we have a 3 prime hydroxyl group. Sometimes either the hydroxyl group will be dropped or the phosphate group will be dropped, but this is still the 5 prime carbon, and this is still the 3 prime carbon exposed. So they have directionality, and we could not have added this nucleotide to the opposite side. The chemistry just doesn't work that way. And if you notice, we added it to the 3 prime end. So nucleic acids, we will talk about this more and more, but they're only able to grow the 5 prime to 3 prime end because on the 3' end, that is the only area where you can add a new nucleotide. Carry the one. Oh, sorry, didn't see you people there. I was just measuring something. Speaking of something, let's talk about DNA. So, starting off, DNA has a deoxyribose sugar in its nucleotide. That means that on its 2' carbon, there is an H. That isn't important to know. Now, this is important to know. DNA can have the nucleotide for DNA can have 4 different nitrogen bases. It can have an adenine, a thymine, a cytosine, or a guanine. Third, DNA has directionality. This is very important. Let's start by numbering our DNA molecule here. We have 1', because we start numbering our carbon on the carbon that is attached to the nitrogen base, where this is nitrogen base, that's thymine. Cool. And this is a 2', 3', 4', 5'. Okay, so on this end, we see that there is a 5' end, 5', and on this side, there's a 3' end, where there's not a phosphodiester bond attached to each. Because DNA or nucleic acids are linear, they have ends. So this would be the 5' end, this would be the 3' end, and we would say that this going from top to bottom, it goes from 5' to 3'. So DNA has directionality. And as you see in this diagram, DNA is double-stranded. It is found in the double-stranded state. But it's not as simple as just slapping another DNA close to it and they stick. They stick in a particular way. They stick in an anti-parallel way. And we can see this by numbering our thing. We start off with the carbon that is attached to the nitrogen base. So this is our 1', 2', 3', 4', 5', 1', 2, 1', 2', 3', 4', 5'. Okay, so you see that this side has that 3'. This is the 3' end, and this is the 5' end. I don't have a lot of room, so I'm going to draw over it. So that means that going from, I guess we'll go from bottom to top. It goes from 5 to 3 on the bottom. So this side is 3', 5'. Okay, we see that in terms of the directionality, while they're parallel to each other, like they're going the same direction, they are reversed. So we call this being anti-parallel. So DNA strands are anti-parallel to each other. Parallel meaning like, they're parallel to each other, and anti, they go in the opposite direction. They are linked in a complementary fashion. And what does this mean? It's like really important. So if you look over here, we see that there are our nitrogen bases, and they seem to be interacting between the strands, and they are interacting via H bonds. Now, so this thymine over here, this representation, it is actually a chemical compound, and so is adenine, and they have different chemical properties. They have different, different polar parts in them that allow them to make H bonds with each other. But a thymine does not usually make H bonds with the cytosine, and nor does an adenine make bonds with guanine. They go T to A and they go G to C. This is what we call complementary. In fact, to define even more, these are called base pairings, because a base would be a nitrogen base and there's a pair. So these are base pairs, and they are complementary base pairs. So complementary is that they link up in this T to A, C to G fashion. And it is also double-stranded in a helical structure, and so it has a helix structure. In the diagram to the right, it looks kind of like this, kind of like a ladder where it goes straight. I mean, they're parallel, and then you have 4 base pairs between it. This only has 3 in them, but whatever. But that is not actually how DNA will exist. It exists in a kind of a winding fashion. So it exists more like this, where it loops and the strands loop over each other. And this makes sense. The helical structure offers further stability to a DNA molecule, and DNA is meant for conserving our genetic material. And the more stable it is, that's good. So the helical structure contributes to that stability compared to if it was just in that ladder structure. So it's in that helix structure. So DNA is anti-parallel complementary in the helix structure. The last thing I want you to know is that DNA is negatively charged. It is negatively charged because it has a negatively charged backbone. So the backbone part is kind of important. So looking at this diagram over here, we see that our base pairs, our nitrogen bases, are kind of like nestled in the center, and like that one and like over here. But exposed to the outside are our sugars, our 5-carbon sugars, and our phosphates. And what do we remember about the phosphate from the nucleotide slide is that they are negatively charged. So this results in going down the entire backbone. Because of these phosphates, the backbone is negatively charged. And also this is called the sugar phosphate backbone because of the sugar in the phosphate. So there is a sugar phosphate backbone that is negatively charged. And as a result of this, DNA is mostly negatively charged. And by DNA having the property, and along with RNA, of being negatively charged, they interact with their environment, they interact with polar stuff, they interact with positive stuff very favorably, and you can do different procedures on DNA and RNA by because of its negative charge. Sorry guys, I gotta measure one more time. I'm just kind of worried. Oh, thank goodness. All right, so let's talk about RNA. RNA, for its nucleotides, they have ribose sugars. You won't need to know this for the exam, but just for funsies, that means that a 2' carbon has a hydroxyl group, which is an OH. What is more important is that RNA has different kinds of nitrogen bases, at least one different one. So the nucleotides can be adenine, cytosine, guanine. However, instead of diamine that we saw in DNA, we have uracil. RNA also has directionality by virtue of nucleic acids being only able to be made in the 5' to 3' direction. We can start by labeling our RNA molecule. Remember that when we're labeling our 5-carbon sugar, our first carbon that we label is the one attached to our nitrogen base. So 1', 2', 3', 1', 5'. Also, I lightly indicated that there is an OH on the 2, and DNA, which has a deoxyribose, it has an H, but RNA has that ribose sugar, which is different in that it has that hydroxyl group. But that's really not important. So 1', 2', 5'. And when I say not important, I mean not important for the exam. So I drew this one off to the side kind of wonky to kind of show that in the 3D space, DNA and RNA and like molecules are always twisting and turning, and they can like turn across a bond. So this is still a valid depiction of it. You can just flip this 180 and then it would be in line with these, but labeling goes the same. We start with the carbon that is attached to our nitrogen base, so 1', 2', 1', 5'. Okay, so we see that on this end over here, we have a 5' carbon, which is attached to our phosphate group. And then we have our 3' carbon, which is exposed and not in a phosphodiester bond as with the other nucleotides. And it has OH on the end of that. So this is our 5' end, and this is our 3' end. So our directionality going from top to bottom would go 5' to 3'. So like DNA, RNA also has directionality by virtue of the chemistry involved in synthesizing a nucleic acid. So what you've noticed is that RNA is single-stranded. So it well, when you produce RNA, we'll talk about this soon, but it is bound to another nucleic acid for a bit, but after it peels off, it is present in a single-stranded form, unlike DNA, which is bound to another DNA strand. And as with DNA, RNA is negatively charged. Each one of these phosphate groups are still negative. So it gives RNA overall a negative charge, which means that it interacts favorably with polar stuff along with positive stuff. DNA and RNA are very involved in each other's processes, so I would like to cover the differences between them. I took the liberty of cataloging the differences that we discussed so far. I hope that's okay with you guys. Anyways, so DNA has a deoxyribose sugar in its nucleotides, whereas RNA has a ribose sugar, and they differ. These sugars differ in what is attached to their 2' carbon position. For DNA, a hydrogen is attached to that 2' carbon, and RNA, a hydroxyl group or an OH group is attached to that 2' carbon. For DNA, its nitrogen bases can include thymine, where RNA does not have thymine. Instead, it has uracil. DNA is found in that double-stranded helix structure, whereas RNA is found in a single-stranded structure. Now, I would like to discuss the implications of this structure because I think that's it'll show like kind of what DNA and RNA are for. So because DNA is double-stranded, it is stabilized by another strand. Each strand of DNA are stabilized by each other with all the base pairs between them, and that helical structure helps as well. So as a result of this structure, DNA is stable. RNA is single-stranded, and as a result, it is less stable than DNA. Now, RNA is a pretty stable molecule. Like, it doesn't really get modified all that much, but compared to DNA, it is a lot less stable. And that's okay because they serve different purposes. Because DNA is so stable, it serves as an excellent information molecule or a permanent information molecule. Like, we don't really want our DNA changing, and by keeping it the same, we can avoid a lot of deleterious mutations, like bad mutations and whatnot. So it is good that it is stable. And RNA is more unstable, but by virtue of being single-stranded, it is a lot more versatile compared to DNA. Where DNA is our primary info molecule, RNA serves a lot of different purposes. It can be used and then broken down and then reused, like the components of it. So RNA could be mRNA. It could function as tRNA. It could function as ribosomal RNA, rRNA, or maybe small nuclear RNA, but those are not relevant to this exam. So the ones we'll be talking about are messenger RNA. And messenger RNA is our intermediary molecule that goes from DNA to protein. And tRNA, or transfer RNA, is the kind of RNA that will help carry amino acids and carry them to the growing polypeptide chain during translation. So long story short, RNA, because it's single-stranded, it can do a lot more. And yeah, it's okay that RNA is a little bit more temporary compared to DNA because it just serves its purpose and then it can be recycled. Let's elaborate more on base pairing, which will tie into nucleic acid synthesis. Let's look at the types of nitrogen bases present in DNA. We have thymine, adenine, cytosine, and guanine. So when you have your DNA, we're going to ignore the helical structure for now. You have your sugar phosphate backbone, and then you have your bases on the inside. These are able to form hydrogen bonds with each other and thus keep the strands connected. But T can't form them with C. I mean, usually can't. This is not favorable. And A is not favorable to join with G. We have base pairs that connect with each other. So for T pairs with A, C pairs with G, A pairs with T, G pairs with C, T pairs with A. So like what's happening all down this line are H bonds. So if you ever heard anything about H bonds, it's where you have a very polar element, like one of NOF. So let's say nitrogen is attached to a hydrogen, create a partial positive dipole up here, and then over here we create a partial negative. And because of these partial dipoles, they attract very heavily to each other. They don't actually create a bond, but they stick to each other a lot. So how I like to remember these pairings is for T and A, they make the word at. And C and G look very similar in shape. A and T, at. C and G look similar. RNA has uracil instead of thymine. Uracil literally just takes the place of thymine. The pairings are the same if you consider U to be T. So you have U, A, C, G. So U would be uracil, and it would pair with A. So you have AU. Now I like to remember this with, that is the chemical symbol for gold. There's also the abbreviation for Australia. And in the same, C and G would pair up with each other. Now, I kept on saying over and over again that RNA is single-stranded, right? So what would be an instance when you actually pair them up? And that would be when you actually are synthesizing the RNA. So the creation of a new nucleic acid, DNA, or RNA, you need a template. So you have this strand of DNA. Let's say that there's an A over here, and there is a T over here, and there is a G over here, and a C over here, and there is a T over here. And the directionality is very important in DNA. So let's say that this is the 3' end, and this is the 5' end. So let's say that you were synthesizing a DNA, how would that go? So I'm just going to show you how it works, and I'll explain it in a little bit. So we would start with the 5' end over here. This is our new strand. And then we would start to make it. And then you have your T. Sorry, you have your T, it goes with that one. You have your A, it goes with this one. Yeah. Okay. So So as you see over here, I started from the 5' end and I worked my way over the 3' end. That is because new nucleotides can only be added to the 3' end. This means that a nucleic acid, DNA, or RNA only grows in the 5' to the 3' direction. Now, the way I like to remember it is kind of like, it's a very roundabout way, but it's the only way I can think about it. I could think of a way to, I don't know, memory device, where would you rather work a 5 to 3 job or a 3 to 5 job? So if you worked a 3 to 5 job, you would literally just work 2 hours and you wouldn't make any money. But if you worked from 5 a.m. to 3 p.m., you can make a decent wage, better than working from, only 2 hours. So we go down the line, we're going from 5' to 3'. There's a C over here, there's a G over here, there's an A over here, and there's the 3' end. So they are anti-parallel to each other. Now this is DNA, and this would be a process of DNA replication. But what added all these new nucleotides? And that was a kind of polymerase. Because this is DNA, evident by the presence of the thymine, this was a DNA polymerase. Now, same would go for RNA. So RNA is also made from DNA due to the process of transcription. So let's say if you have a 3' to 5' thing over here, and then it has an adenine, a thymine, guanine, cytosine. Yeah. So you start from the 5' end. It is always replicated 5' to 3' because you'd rather work a 5 to 3 job than a 3 to 5 job. So you have a polymerase that will add these nucleotides on, and this is an RNA polymerase because we are synthesizing more RNA. So we think about, okay, what pairs up with our A? A thymine, but this is RNA we're talking about, so it's a uracil. And then this thymine, adenine is totally good. You can make a cytosine, you can make a guanine. Yeah, and we synthesized it in the anti-parallel direction and going in the 5' to 3' direction. So going forth, whenever you are talking about nucleic acid synthesis, it goes anti-parallel to template strand, which would be your DNA. And it is only synthesized, made in the 5' to 3' direction. And earlier, just in case you're curious, like with the RNA being duplicated by the DNA, there are different ways that you can peel the RNA off of the DNA. This is the RNA template that you use to make your RNA. This is the DNA template you use to make the RNA. The RNA can peel off and separate and then become single-stranded again. So RNA is never like really bound to another RNA molecule, but it can exist in a double-stranded form when it is being synthesized. A prokaryote is a cell that has no organelles. This means no nucleus, no mitochondria, no chloroplasts. Eukaryotes are cells that do have organelles. The one we'll be examining is the nucleus. So in your eukaryotes, you have your nucleus, and then housed within your nucleus is all your genetic material. Within prokaryotes, your DNA is just like, it's just spread around. It's not enclosed by anything particular except the main cell wall. Or not cell wall, the main cell membrane. And this has a number of effects. Eukaryotes are generally capable of greater levels of sophistication, and they can regulate their DNA more, they can modify their RNA, and that is the main difference that we're looking at. Eukaryotes have RNA modification. In reality, transcription, translation, and even replication are extremely different from prokaryotes and eukaryotes, and I don't think they even touch on this in class, so you do not have to worry about it. So know that eukaryotes, because they have that nucleus, are capable of RNA modification, and that, okay, so there is a term that cytoplasm. Cytoplasm is everything within the cell, housed within the cell, but nucleus. So the prokaryote doesn't even have a nucleus, so that's just everything in the cell. And then for the eukaryote, it would be everything in the cell except what the nucleus and what is inside the nucleus. So any process that a prokaryotes have happens within the cytoplasm just by default, because there's nowhere else where it can happen. Whereas for eukaryotes, within the nucleus, that is where transcription happens. Okay, so what I want you to take away, eukaryotes have additional RNA modification. They do transcription within the nucleus because that's where their DNA is. Whereas prokaryotes, they do it within the cytoplasm because the cytoplasm is everything within the nucleus. I mean, within prokaryote, it's like 4 o'clock in the morning.